Happy snow day, ladies. Today we're going to keep moving forward in your notes, learn a little bit more about Gregor Mendel and what he did, and sort of some basics in predicting inheritance. So, if you remember last time we left off saying that Mendel was going to study 7 different traits, each of which he believed to be inherited in an either-or fashion. And so what his goal was to give evidence that you don't get blended inheritance in living things, but rather you inherit one trait or another. And he believed that you could predict how this would happen. So Mendel crossed plants that were true breeding for each of his 7 characteristics. And the way he did this is crossed a true breeding with one version of the trait with a true breeding that had the other version. So as you can see in this chart, he crossed a round pea plant with wrinkled peas plant. He did a yellow seeds with green seeds, a gray seed coat with white seed coat, and so on and so forth. Now, the original pair of plants he referred to as his pea generation or his parental generation. And the offspring that came from those plants he referred to as the F1 or first filial generation. So, as you can see in this table, when he did these parental crosses, he got F1 generations that showed all one trait. So when he crossed round and wrinkled peas, he got all round offspring. When he crossed yellow and green seeds, he got all yellow offspring. When he crossed smooth and constricted pea pods, he got all smooth pods. And so he showed right away evidence that you didn't get a blending of inheritance, you actually got one type of the trait or another. Now, he began referring to the F1 offspring as hybrids, a term we hear a lot today for various reasons, a hybrid being a mix of things. And these F1s were hybrids because they were genetic crosses between plants with different traits. Now, even as hybrids, they only expressed one trait. So again, smooth pods and constricted pods formed a hybrid child, but the hybrid offspring showed smooth pods. So again, he showed that the blending was not taking place here. So after doing several of these parental crosses, Mendel came up with 2 conclusions that he wrote about. The first one is that inheritance of traits is determined by what he called factors that pass from one generation to the next. Now, obviously today we have a different word for factors. We call them genes. We know that they're segments of DNA that serve as the instructions for how to get a particular characteristic in an organism. Now, we also know today that genes come in one of 2 contrasting forms or versions, and we call these alleles. So basically, you inherit alleles that determine what your characteristics are. So, like I put on this slide here, the trait of plant height, you could have a tall allele and a short allele that could be given to your offspring. And so for every trait, an organism will carry 2 alleles in some combination. These 2 alleles would be one from your mom and one from your dad when we talk about sexual reproduction, and we'll get into that a little bit more later on. Now, the second conclusion that Mendel made is what he referred to as the principle of dominance, the idea that some alleles will be dominant and other alleles will be recessive. And this is one of the things that I hope you've kind of heard of before in science classes. A dominant allele will code for a trait that will always show up and be exhibited if you have one of those alleles. So even if just one of your alleles is dominant, you will get the dominant characteristic or the dominant trait. A recessive allele codes for the other version of the trait, and it only shows up if that's the only thing you have. So if you have 2 of these recessive alleles, then you'll see a slightly different characteristic. This is how Mendel explained what he saw in that F1 generation. all of the characteristics that showed up in those offspring must be the dominant traits. So again, when he crossed a tall plant with a short plant, he got all tall plants. So he determined that tall must be the dominant trait. Now the way we write things is dominant traits are designated with a capital letter. And it doesn't really matter what letter we use, but we always try to use letters that it's easy to tell it's easy to tell a capital from a lowercase. So for instance, you don't usually want to use S's, right? Because a capital S is kind of hard to tell from a lowercase s. So we usually choose a letter that's easy to tell the capital letter. And we also usually choose a letter that makes sense. Like you see here, we use a capital T for tall, a capital R for round, a capital Y for yellow. Then the recessive trait that matches that, we would designate the lowercase same letter. So for instance, if capital T is tall, then little t is short. If capital R is round, little r is wrinkled. If capital Y is yellow, little y is green. So if we consider one of the pea plants from Mendel's examples, right, let's say the height trait here. The dominant allele would be tall, capital T. The recessive allele would be short, lowercase t. So in this picture, we know that this tall plant has a big T and this shorter plant has a little t. But every plant cell should have two alleles, one from their mom and one for their dad. So we can get one of three possible combinations of alleles. We can get two big T's, we can get two little t's, or we can get one of each. Now Mendel wasn't satisfied, though, with just a P generation and an F1 generation. Because if Mendel's idea of dominance was true, then it's possible that these recessive traits were kind of hiding here, right? And to really show evidence that he was right, he would need to see another generation that he called his F2 generation, and he would expect to see that maybe those recessive traits would come back in this generation. So, what Mendel did is he crossed two of his F1 plants. So if you follow this picture, he had his P generation here, a true breeding tall plant and a true breeding short plant. Then he had his F1 generation here, which were all tall. And he crossed these two plants with each other. So one of these F1 tall plants with another F1 tall plant. What he saw is that in this third generation, or this second filial generation, the F2s, he saw that you got three tall plants for every one short plant that showed up. In other words, a 3-to-1 ratio. And this was evidence that the recessive trait could reappear in these offspring. Now, the fact that recessive traits came back in that F2 generation showed Mendel that sometime in inheritance, the two factors you have, the two alleles that you have, must separate or segregate from each other so that they could be individually inherited in the different plants. He called this his law of segregation. Now, today's understanding is that this segregation, this separation of alleles takes place during meiosis, which is critical before we can do sexual reproduction. Now, we talked about chromosomes some in the first semester as we came to the end. You have homologous chromosomes in your cells, chromosomes that are really similar to each other. And what we know is that those two things will separate from each other during meiosis. Now, we'll get into this a lot more later, how this works and things. But the idea is each parent might give you one allele or the other allele that they are carrying. So due to this idea of alleles separating, what we can imagine is that when the body forms sex cells, gametes, right, sperm and eggs, each one of those will actually just contain one of the possible alleles that you could get from your parent. So like, let's say that the parent plant has big T, little t as its two alleles. That means that the gametes this plant makes will either have the big T or the little t, and that's what will be passed on to its offspring. So this will make better sense once we talk about meiosis a little bit, but a cell will make 4 gametes at once, and when it does this, it will make an even number of gametes with different alleles. So it could give a big T in its gamete, or it could give a little t in its gamete if it starts out with big T's and little t's as its alleles. And then these gametes are what go on to form the new organism, or in this case the new plant, and will carry with them either the information to be tall, if it carries from a big T gamete, or short, if it comes from a little t gamete. Now, the result of this entire process is going to give us a new generation of plants, or offspring, that have new combinations of alleles. And what scientists were really most interested in is, when you look at parents, can you predict what we'll see in their offspring? Now, the answer is yes, you can. And the way that we do this is through mathematics, right? Probability. Probability uses math to try to tell us how likely is it that a particular event will happen. And usually what we're talking about here is how likely is it that an event will happen compared to another event. Now, even if you're not a math person, don't freak out here, because probability and the way we use it is going to be very simple. And the reason it's going to be very simple is because we really just have 2 possible outcomes, right? We can either get dominant or get recessive. So probability in this case is basically like a coin flip. If we toss a coin in the air, right, and we flip a coin, there's 2 possible outcomes, right? Heads and tails. What are the odds that a coin flip will turn up heads? What are the odds that a coin flip will turn up tails? I'm confident that you know the answer to that, so let's keep going. So when we flip a coin which has heads on one side, tails on the other, we essentially have a 50-50 chance of getting heads and a 50-50 chance of getting tails, right? Now, we can then write that as a probability, and probabilities can be written in one of 3 different ways, right? You can write a probability as a fraction, right? So what are the odds that we are going to get heads? As a fraction, that would be 1/2, right? Because half the time we would expect a coin to land heads up. You could also express this as a percentage, which would be 50%. And again, that's because 50% of the time we would expect the coin to land heads up. Or we can present this as a ratio. Now, a ratio is where sometimes students struggle a little bit. If we write this as a ratio, the ratio is actually 1 to 1 for a coin flip. In other words, for every time it lands up heads, we would expect that there would be a time that it lands tails up. Now, a lot of times what people want to do is they want to write this ratio as, they want to write 1 to 2 for this. And that's because they're used to writing in fractions. But this is not correct, right? When you look at a ratio, the sum of your numbers, 1 plus 1, should be the total possible outcomes. And so with a coin flip, again, there's 2 possible outcomes, heads or tails. So the numbers in our ratio should add up to 2. And so we say that it's a 1 to 1 ratio. Now, honestly, in my class, you can use any of these that you're comfortable with. If you're comfortable with ratios, great, you can use them. If not, err on the side of caution, use percentages or fractions. Now, also realize that on a coin flip and in genetics, past outcomes don't affect future outcomes. So if I flip a coin and it lands heads up, the odds of heads or tails on the next coin flip are still the same, right? They're still 50-50. Even if my coin, even if I flip it 50 times and I get heads every single time, that 51st flip, heads or tails is still equally possible, right? And this is the same way that it works in genetics. Each reproduction event is its own new event. And so the outcome of other events do not affect the one that we might be studying. Now, because alleles separate randomly, just like when we flip a coin, we can use the exact same principle to try to predict what our inheritance outcomes would be. So let's take an example, break it down, and look at what might happen with pea plants in terms of that inheritance, and we'll see if it fits what Mendel saw in his experiments. So first, a couple of things. Expression of alleles and some vocabulary here. So the allele for a dominant trait is going to be a capital letter, in this case big R, for round, right, in these guys. So big R equals round. If that allele shows up at all, even once, then our P's will be round. But if it doesn't show up and we're left with just little r's, then our P's will be wrinkled. So if a pea plant got big R, we would say that it would have round P's. If a plant got big R, there's still a big R, so we would say that it also gets round P's. But if it happens to get little r, then there's not any capital R's, and we would say that it gets wrinkled P's. Now this leads us to two really important vocab terms. Really important. And here's why they're important. I'm not going to ask you these terms specifically, most likely. Right? I'm not going to ask you on a test, what does genotype mean? But I'm going to ask you in a genetics problem, what are the genotypes? So you have to know what that word means to be able to appropriately answer that question. Or I'm going to ask you, what are the phenotypes? You've got to know what that word means to be able to answer that question. So, genotype. Genotype is the genetic makeup of the cellular organism. So it's what genes they have, right, or what alleles they have. So when you're asked for a genotype, you're going to give me two letters, right? So like from the last slide, big R, big R, little r, little r, those are all genotypes. Now then, the genotype tells you what the phenotype of the organism should be. The phenotype, remember this pH here, is the physical expression of alleles. So the phenotype is what characteristics the plant should have based on the genes it gets. So for instance, round and wrinkled from the last slide, those are phenotypes. I was really struggling not to go ahead and just say that word on the last slide. So you have a genotype, which are what alleles you have, big R, and you have a phenotype, which is the characteristic that gives you. So big R gives you round seeds. Alright, so another way to kind of check yourself is a genotype should just be a couple of letters. A phenotype should be a descriptive word, right? Round, wrinkled, yellow, tall, whatever it might be. Now, a couple more important vocabulary terms. If your genotype consists of the same type of alleles, so could be big T, or could be little t, doesn't matter what letter there, right? If you have two capitals or two lowercases, we say that you are a homozygous individual or a homozygous cell. Homo meaning same, right? So you can either be homozygous dominant and have two capital alleles, two dominant alleles, or you can be homozygous recessive and have two recessive alleles, two lowercase letters. If you are that third possibility, one big, one little, then we say that you're heterozygous, hetero meaning different, right? So heterozygotes or heterozygotic cells have one capital letter, one lowercase letter. All right, so here's a picture of me being a square, right? To kick off, Punnett squares. The one thing in biology that a lot of people always say they like doing, I don't know why that is, and you might disagree with me, but that's one thing I've always heard from students is, ooh, I like these. I remember these from last year. These are fun. And we're still learning. So hopefully you'll find that they aren't too complicated. I think that's why students like them, because they're not very hard. But it's a great way to visualize probability, and that way not think about it so much as math, but just look at it like it's a little picture. So a Punnett Square was devised after the time of Mendel. That's why they're not called Mendel squares, right? Other scientists did this. But these are squares that we can use to predict the outcomes of a cross between two organisms. And this prediction is a little simpler when we use a diagram versus if we just try to do it mathematically. Mendel basically did his predictions just with the math. These guys came up with a simpler way to do it by drawing a little picture. Now, the most common mating event that we consider is a monohybrid cross. Mono meaning one, right? So this is where you form a hybrid, and you're looking at the cross of just one particular trait. So like height or color of the seed, etc. Now a Punnett square for a monohybrid cross is drawn as a simple 2x2 grid, right? A little 4 square. And what we do is we write the parental alleles up here at the top and here on the left-hand side. And then we use this grid to try to predict what kind of offspring they would get inside these boxes. Again, I'm hoping that many of you have done this before, but just in case you haven't, we're going to start from the ground and work our way up. So let's consider one of Mendel's original P generations. If he had true breeding plants that he crossed, that means that he crossed a homozygous dominant plant with a homozygous recessive plant. So one parent would have been homozygous tall dominant, we'll say big T, meaning that it could give its offspring either a big T or a big T, right? Now the other parent would be homozygous recessive, short plants, little t, meaning that they could give their offspring either a little t or a little t. Now here's how we'll handle this with a Punnett square. To set up your Punnett square, you put your parental alleles at the top, right, and down this left-hand side. And then to make your predictions for the offspring, you realize that every offspring should end up with 2 alleles, one from the mom and one from the dad. So what you do here is what I refer to as crossing and dropping. Sounds kind of like a hip-hop dance, doesn't it? Cross and drop. So the way it works is you're going to take your parental alleles from the top and you're going to drop those down. All right, so I'll put a big T here, I'll put a big T there. We're going to drop this one down. Big T there, big T there. Then you're going to take your alleles from the side and you're going to cross with those. So we're going to put a little T here, little T here, little T here, little T here. Now again, remember what Mendel did, right? He crossed the 2 true breeding parents up here and over here. And then in that next generation, he saw all one trait. So here we get all of our offspring, 100% of our offspring should be heterozygotes. And those heterozygotes, because they have a big T, would all be tall. So this matches up with what Mendel saw. Now what you're often going to be asked is, okay, do this Punnett square, what's your genotype outcome and what's your phenotype outcome? So for what we saw on the last slide, 100% of these plants will be heterozygous, big T, little t, 4 out of 4. And 100% then of those plants will be tall because they have that big T. Now, let's continue on with Mendel's experiment. The next thing he did is he crossed 2 members of that F1 generation. So now our parents are a heterozygote from the F1 generation and a heterozygote from the F1 generation. And so again, let's cross and drop. Okay, we're going to drop our big T. We're going to drop our little t's. We're going to cross with our big T. And we're going to cross with our little t. Now notice what I do here. It's not a hard and fast rule, but tradition, convention, they show us that you always write the capital letter first. So a heterozygote, for a heterozygote, you never write little t, big T. All right, you write big T, little t. And so I made sure that I came out that way in my Punnett square. So notice what I get this time. a mixture, right? Not all one genotype and not all one phenotype. So let's take a look on our next slide here at these outcomes. So again, I'm going to draw this Punnett square really quickly up here at the top so we can reference it. Get big T there, big T there, big T, two little t's. Okay, so if we're asked about the genotypic outcome in this cross, this is F2 generation that we form here, we're going to find that 25% of our plants are homozygous dominant. We're going to find that 25% of our plants are homozygous recessive, and we're going to find that 50% of our plants are heterozygous. Now, how does that translate into phenotypes? In terms of phenotypes, 75% of our plants will be tall. That's both the green and the red ones that I circled above. And 25% of our plants would be short, which are the blue circles above. Now, in other words, if we want to put this as a ratio, we say that we have a 3-to-1 tall to short ratio. 3 tall for every 1 short plant that we would get. And again, this is exactly what Mendel saw with his true evidence. When he crossed the parental generation, he got all the same for the F1s, but when he crossed the F1s, he got a 3-to-1 ratio of dominant to recessive. So to wrap up, the last thing I want to point out and make sure you realize is that probabilities are predictions, and they're predictions of averages. They're not necessarily true outcomes, and they're not necessarily a certain prediction. So a Punnett square is a great way to predict what will happen, but it won't exactly tell us what will happen. An example for you, if Punnett squares were perfect, then every single family that we looked at would have an even number of boys and girls, assuming they had an even number of kids. So if you had 4 kids, you should have 2 boys and 2 girls, if Punnett squares are always true. And you guys probably know from your own experience that that's not the way it turns out, right? Lots of families have all girls. They have 3 girls, or they have 3 boys, or whatever it might be, 2 boys. So Punnett squares are great for predictions. They're going to be very helpful for what we're going to do in class, but remember that they are not certainties. And also, realize that the more data you have, the better. So the more times we produce offspring, or you can think of it this way, the more times we flip a coin, the closer we're probably going to get to our prediction. Right? In other words, the better our data is. Right? And we've known this since the very start of school. The more data we have, the better. And we're probably going to get closer to our expected values, closer to our predictions, if we have more data. Okay, so this takes care of where we need to get for the first part of Chapter 11. And now you guys are going to start practicing some Punnett square problems for homework, some of which will be graded, and some of which will really just be practice that you'll work on yourself, and then we'll go over in class. Have a great day.